So is that screen visible to you? Can I start? You can start, right? Okay. So good afternoon, students. I'm Dr. Ramkrishna from SRM University. Today I'm going to teach you two topics. One is flow cytometry, and second one is light therapy. So these two topics has a common point, which we are actually using either a light source, which is a LED or laser, in both the technologies like flow cytometry technology and light therapy technology, and they both have different applications. Am I audible? Am I audible, Alvin? Right. So this flow cytometry and light therapy applications are completely two different applications, but the common thing between these two is usage of light. Light as a sense, either a laser or high intense LED beams. So these are the things that we are going to use it for a particular applications like flow cytometry, and second one is light therapy. So let's discuss one. The first one is a flow cytometry, which is a very important analytical tool for various applications, ranging from pharmaceutical industry to biomedical industry, and of course the biotissue engineering, and pharmacology, and the forensic department. So there are so many people are actually using this flow cytometry technique. So what is this actually flow cytometry? As it says in English, flow is actually a movement. It's some particles are moving, and cyto means actually the a particle, which is a cellular body or any entity. A cyto means it's like a particle or any entity, and metry, of course, is a measurement. So when some cellular body is actually moving, it's actually moving in either a air medium or maybe a liquid medium. If you are able to analyze their size and its composition of chemicals or any other parameters, then it's called cytometry. Flow cytometry means when it is in a movement. So let's look at how this flow cytometry looks like. This is a very a typical structure. You can just Google it. It is available in Google also. It's a very simple diagram to understand what is flow cytometry. So you see that you can see here, it is actually a simple nozzle. It's a simple nozzle. You can see that, and you see that there is actually a fluid that is coming in. So this is called sheath fluid. This will actually take your molecules. This actually works as a vector. That actually works as a vector. Of course, a guide to route the molecules which are actually coming from the sample injection. So from the sample injection, if you give any molecules, for example, cellular bodies, maybe a white blood cells, RBC, or maybe some chemical composition molecules, or maybe nanoparticles, or maybe any kind of cellular mechanism, or maybe a bacterial elements, whatever is that, you're actually giving that as a sample here, or maybe a human cells, right? So whatever that you are actually giving here need to be routed properly through this column. So for that, we are actually using the sheath fluid here, which could have some particular pH to maintain the a proper flow. And we don't need to create any turbulent flow here. We should not create any turbulent flow. It has to have laminar flow, so that the sheath fluid will help you to take your particles into this nozzle and creating a laminar flow here, and not have any disturbance in flow pattern. And you see that the samples are actually coming from top, and these samples, there are many samples. It's a heterogeneous samples, which means that there are different sizes of samples could be there, are different types of entities, for example RBCs, WBCs, and other blood constituents, or maybe a plasma, all together actually coming into this particular line. And this is being guided slowly. These particles are guided slowly one after other through this needle like structure, and then it is passing through. And what we do now, there are possible measurements like there are size measurement or composition measurement, chemical composition measurement. All these are required, right? So we should go with the light based technology, which includes every cell has its own property, light properties, which means that absorbance and scattering properties. And of course, that absorbance and scattering properties merely not depending on the just cellular body. It is highly depending on the composition of the cellular body. Many compositions, for example, carbon based elements could be there, sulfur based elements, or maybe nitrogen based elements, oxygen based elements, and there are so many other chemical composition also there. So the absorbance and scattering properties of these particular entities which are coming here is completely different and of course unique for that particular structure. For example, I have a protein content, I have a DNA content, I have a red blood cell, I have a hemoglobin, I have, there are so many entities are there, and their compositions, chemical compositions are completely different. So passing this laser light through this nozzle helps us to understand the absorbance properties and the scattering properties of this particular body, cellular body. So let's see the principle of flow cytometry, how it is looking like. The basic principle of flow cytometry is the passage of cells in a single file in front of a laser so they can be detected, counted, and sorted too. So sometimes you can actually sort the entities also by light force itself. So the next one is the cell components are fluorescently labeled. So each cell, if you can, sometimes what happen, if the cell inherently absorb light or inherently scatter light, you don't need any external molecule. But here sometimes your molecules or the cellular bodies may not have the absorbance or scattering properties in a particular wavelength region. So to identify that, we wanted to take help of fluorescent molecules. So it is like a dyes where you're actually using the fluorescent dyes, so and having some specific binding nature to the cellular body. So if you use this fluorescent dyes, it will go and bound to the cellular surface, or sometimes it will go inside the cellular cell. It is actually penetrated to the cellular surface and going into the cellular body. It may be reacted with mitochondria, or it may be reacted with the DNA, or it may be attached to the protein content available in the cellular body. So you see that every cellular body, for example, like lipid molecules are there in the cellular body. So lipid specific fluorescent, if you are using, the fluorescent molecules are sitting onto the lipids, which is the cellular wall, that is called cellular wall. If you are using the fluorescent molecules which is very specific to the protein content inside the cellular body, then that molecule is actually penetrated inside the cellular body, and then it will react with your particular protein structure, maybe an antibody or antigen or so. Sometimes it may be very specific to the DNA compounds, where adenine, glutanin, and there are other 4 ATDC molecules are there. So it will be going and binding to that particular molecule. So what is so important with using this fluorescent technology? Because these fluorescent molecules, you can tag it to a particular antibody or tag it to a particular targeted element, and then if you use it, so the cell compounds, one single cell components also can be visualized. You can actually see them through your microscopes and other light based technologies. So this is very, very important. The flow cytometry here, it is actually a flow of cellular organs, which is flow means motion of cells. Flow means it's a motion of cells. How it is moving? It is moving in a soothe fluid or the sheath fluid, and it is being guided to a particular nozzle. And cyto means a cellular body. It can be a human cell, it can be a plant cell or animal cell, or maybe a bone marrow cell, or it could be any particular cell. So that particular cells are actually rooted here. And metry means measure. So measuring properties of cellular body in fluidic environment is called flow cytometry. So I hope you understand this technique. So this technique is very, very, very simple enough. However, there are three different things are actually there. One is the flow control unit. It is first one is the flow control unit, which means the flow dynamics and flow, how the flow has to be moved, how this cellular particles has to be distributed. All these things is one particular physics of it. Second physics is laser and its interaction with the cellular body. So flow control is completely a one particular dynamics, and laser interaction is also another particular physics. There are two physics involved in it. After that, electronics part is also there, because your light which has been scattered over the molecules or the cells, or maybe either it's a forward scattering or relay scattering, or maybe fluorescent scattering or whatever is the scattering, it has to be detected by photo detectors or photo multiplier tubes, and after that it will be given to the electronic boards, I mean ADCs and other electronic boards, to read the count, how many fluorescent particles have been passed through. It is not just how many fluorescent particles. Fluorescent particles are actually attached to the molecule, and it will give you some absorbance value. So looking at the absorbance value, after taking the curve, so after taking the curve, you can actually find out how many number of particles are there. Barely you can find it out. So let's look at it. Flow cytometry is a widely used laser-based method for analyzing the and expression of cells, surface, and intracellular molecules. You understand, right? Generally, how many cells are there? If that is the thing, you can go for the count itself. Otherwise, intracellular molecules, for example, proteins, DNAs, mitochondrial structure, any other carbon-based structures are there. All of them can be detected over there. There are many applications of flow cytometry in research. So, what are those applications? Let's look at it. One is the diagnostics purposes. For example, I wanted to find out a cancerous cells in a particular human body. So we generally collect the bone marrow, or maybe the blood sample, or maybe the tissue sample. Biopsies cases, we are actually collecting it. Sometimes you see that in forensic department, people used to collect the tissues from the dead bodies, and they wanted to find out whether any particular chemical component is available in the tissue, or maybe the tissue is cancerous or not, or is there any other particular molecules have been introduced. So all that information can be you get it over by flow cytometry analysis. So this is for diagnostic purposes. And second one is including the simultaneous and multi-parameter analysis of a single cells. For example, in a single cell, how much protein content is there, that also you can find it out. And characterizing the and defining the different cell types and heterogeneous cells population. For example, I have a hemoglobin inside a RBC. How much percentage of hemoglobin is there? Every RBC will not have the same amount of hemoglobin. RBC carries hemoglobin is fine, but each cell will carry different amount of hemoglobin, and that can also be find it out. How many cells that are carrying low hemoglobin content, how many cells are carrying high hemoglobin content, can also be detected. Not only that, heterogeneity means you can have different molecules also can be detected at a time, or same molecules having higher or differential amounts of particular amount of chemical compound is also be detected. The next point is flow cytometry has, I mean, that assays can also assess the purity of isolated subpopulation, which means that, for example, so many examples in the pharmaceutical industry, maybe we are actually preparing a drug, and the sediment of that particular prepared drug, I need to find out how much is the content of it. So the subpopulation, which means that you can actually analyze different sizes and different types of molecules and how many of them are there in a given liquid. So subpopulation also can also be detected. Analyze the cell size and volume. So that is very, very important. Cell size and volume can be analyzed, and sort different cell population. So two different cells, you can actually sort it, two or three or many can be sorted out, known as fluorescence activated cell sorting, FACS. This is a fax. So fluorescent activated cell sorting is one big technique which has been utilized in our biochemical laboratories, pharmaceutical laboratories, and drug industry, and forensic department. People are actually using them. And coming to the flow cytometry test measures, what is it actually measuring? Can anybody tell me what is it measuring? Flow cytometers are actually measuring fluorescence intensity. Please look at it. It is very important. Fluorescent intensity produced by the fluorescently labeled antibodies specific to the proteins, or in a cell, or ligands that bind to the specific cell associated molecules, such as propedium, iodine binding to DNA. So it's like, you will be taking a fluorescent molecule, it is attached to an antibody. You can see that antibody structure, it looks like a Y structure, and a fluorescent molecule is attached to the antibody, and this antibody can actually move, and it will bind to either the cell surface where the peptides are there, or inside the cell if there is a particular protein body, or maybe you can go with DNA molecules. So any specific thing that the antibody is being designed, so it will be attaching to that. So once it is attached to that, and a particular antibody is being attached to a cell surface or internal molecules, then you can actually measure how much fluorescence is actually coming out. So taking a graph of fluorescence for a given known amount, then we can actually look into how much is being the amount of the cell or the volume of the cell and size of the cell can be associated. It is not just for accounting and finding out the association of the elements. You can also image it. Fluorescence imaging is also there. Flow cytometry based fluorescence imaging is also one of the another technique. So the staining procedure, it is very, very important. How do you stain? Sometimes your cell body may not have inherent absorption or inherent scattering for a given particular wavelength. In that case, generally we go with staining molecules, which is fluorescent molecules. So the staining procedure involves making a single cell suspension from cell culture or tissue sample. Very, very important. Staining procedure is involving making a single cell suspension. So you just wanted to take out only a particular cell. For example, RBC I'm targeting. So single cell suspension from cell culture or tissue sample. So you have a cell culture which has been into the Petri dish, or tissue sample if you have taken. In the tissue sample, how much RBC is being there, or maybe it's a blood tissue or blood sample, how much RBC is there. So if you're taking the human tissue, what could be the DNA amount. So if you wanted to target a single cell suspension, you can actually go with a staining procedure for that particular cell. So then the cells are incubated in a tubes or micro titers plates with unlabeled and fluorophore labeled antibodies and analyze on the flow cytometry. So you can take unlabeled versions and labeled versions, and you can actually visualize them, count them using the fluorescence scattering. So multicolor fluorocytometry is also available in the market right now. So if you wanted to target two but different entities, for example, RBC hemoglobin I wanted to target, and maybe I wanted to target the DNA molecule. Hemoglobin and DNA, they are two entities. So now I can target both of them with differently fluorescent molecules or the dyes. So multicolor flow cytometry also very, very, very important for us. So it is also available in market. So in multicolor flow cytometry, fluorescent markers are used to characterize and define different cell types of interest in heterogeneous cell population. So in a heterogeneous cell population, you either can go with two cells, two different cells. I wanted to find out beta cells and alpha cells, like, and I wanted to detect differently. You can detect using the two dyes, and you can actually use for isolated subpopulations, analyze cell size and shape. That is all possible in multicolor flow cytometry. So let's look at it, how this flow cytometry works. So flow cytometry allows the qualitative and quantitative analysis of several properties of cell population from virtually any type. So virtually any type of the fresh unfixed tissue and body fluid. So you take any fresh sample, which is body tissue, bodily tissue, or maybe a fluid, for example saliva, or maybe your urine samples, or maybe your plasma sample, blood sample. You take any of the samples that you have collected from maybe a site. So now the properties measured includes a particle's relative size. Generally we are looking at the cell size and relative granularity, what is actually, how the distribution is there, the cell, or internal complexity. For example, what could be the mitochondrial structure inside, what could be the DNA orientation over there, and then that and all you can. How much is the DNA content available in the cell also can be done. And relative fluorescence intensity, how much fluorescent intensity is coming up, it will tell you the amount of DNA or amount of protein that is been inside. So most common analyzed materials are blood, bone marrow, bone marrow aspirate. So generally for cancer, bone cancer people, or maybe CMT related issues, some cancer issues are there. So generally people collect the bone marrow and try to see whether you are able to produce the enough RBCs for circulation of blood and carrying of oxygen or not. So such cases also people have been utilizing the flow cytometry. So you look at this structure, you see I told you there are three important things. One is the nozzle here, where it is actually talking about the flow pattern, where you are actually controlling the flow pattern that is being given here. And another one is the laser part, which is coming it is given in the other side. So this laser is illuminating the cell or multiple cells. Generally it is a single cell, or sometimes it is a multiple cells. So it is illuminating it. Once the illumination is happening, there is a one immediate thing is the forward scattering. This forward scattering, FSE, forward scattering, is been collected by the photomultiplier tube here, and it is converted to the electronic signal, and it is given to the ADCN for processing. And after that, we wanted to look into the fluorescence ejection, that is called fluorescence ejection emission. in different orientations. We are going to collect it for different orientations, and wanted to use the different filters. For example, I wanted to collect the green color fluorescent emission, the red color fluorescent emission. So they are going to be having some dichromatic mirrors, and we'll be collecting what is the amount of different wavelengths that is being emitted over there. So all of them that is being collected and converted into the electronic signal. So this is called scattering, secondary scattering, or spontaneous scattering signals. So this is also being collected. So all of them are actually collecting the light which is being ejected by the, or scattered by the cell that is being present under laser. So either it is absorbing and emitting light, or it is simply scattering light. So all of them can be analyzed over there. So look at it, the first one, the fluids part. So look at the fluids part. The nozzle is being designed such a very nice way. You see that there are two entries are there. So around it, it is like a concentric cylinder. So in the center one, the first cylinder inside, the internal cylinder where you're actually injecting your sample, which is a tissue sample or blood sample or so, having different cells inside. So the other external side will actually have a sheath fluid, that is will be guiding fluid that will take your cells into the nozzle. So central core through which the sample is injected, outer sheath contains a faster flowing fluid. So sheath fluid, it is called sheath fluid, and enclosing the central core. Once the sample is injected, they are forced into the center of the stream, forming a single file profile. So once you're actually pressing the stream is very fast in nature, so it is actually dragging your molecules into the single streamline. And of course, this will have a laminar structure. The flow pattern is a laminar structure inside. There is no turbulence inside. So when there is a laminar structure, the flow pattern is the highest amount of velocity is in the center, and around it is very low. The velocity is being reduced. At the walls, velocity is 0, and at the center it is high. So you see that the pattern looks like it is actually dragging your molecules one after other into a straight line pattern over there. So second part is the optics. So you know that we can shine the light onto the particle. So how you're going to shine the light? There are options for us. Generally, we go for lasers, more commonly used for this kind of applications. But before inventing the lasers, arc lamps are available. For example, sodium vapor lamp, or any other, like ruby lasers or something like that. There are different lamps can be utilized for fluorescent photometry also. However, when you are utilizing the lamps, you have to highly rely on the optical elements, like convex lens and concave lens, or maybe cylindrical lens. There are other lenses are required in such cases when you're using the LEDs or arc lamps. But when you use lasers, it's more efficient ones to focus the light onto a particular site of interest and try to understand the elements over there. When a light inserts a laser beam at the so-called interrogation point, this is called, the light is being focused onto a particular point, is called interrogation point. Two events occurs when the light is being falling onto the interrogation point. There are two things can occur. One is the light scatters, it is actually scattering from the surface, or emission of the light, it is being absorbed by the fluorescent molecules and it is releasing a different wavelength light. So fluorescent molecules absorb a particular wavelength of light, and then it will release a different wavelength of light. It has been well known for you, Raman and anti-Raman, Stokes and Raman concepts are there, where you're actually absorbing light and emitting light. These concepts have been known to us. So now you look at it, the light scattering in two fashions. The first is the forward scattering, the second one is the side scattering. So forward scattering, generally whatever the laser beam that you have, that laser beam is moving ahead, and you see there is a reduction in the laser intensity. Initially, without having molecule over there, the laser intensity is, let's say, 100 microwatts. If the molecule is there and laser has passed through, 10 microwatt or 15 microwatt will be absorbed by the molecule, and the additional amount will be, so the other amount will be falling onto your photomultiplier tube, and you can read 80 to 90 microwatts over there. So there is a forward scattering effect. Second one is the side scattering, where light, whatever is being absorbed, it will actually absorb by the molecule and re-emit by the molecule at a different wavelength. And now that is called side scattering, where we wanted to collect the side scattering using some optical lenses and and direct it to the photomultiplier tubes. Of course, these optical lenses having different chromatic mirrors are there, and these chromatic mirrors or chromatic isolators will transfer the light into different components over there, and then we can find out what is the each component value. So emission of light is the fluorescent molecule present in particular interrogated by the laser light. It will absorb energy from the laser light and release the absorbed energy at a longer wavelength. Emitted photons pass through the collection lens and split into sheared down, and you can actually find it out. So how the data analysis has been done? So the data are usually presented in the form of single parameter. For example, it's a dot pattern, or maybe a histogram pattern, or maybe a contour pattern, or maybe cytogram pattern. There are different patterns you can produce the data, number of molecules of a different size, the distribution you can actually put it up. There are two different molecules, that distribution also you can actually put it up. And the last one, the histogram pattern, is very, very important. This will tell you how many number of particular size you can actually identify over there. This kind of data is been plotted and one can utilize it. So this is about your flow cytometry. So in the flow cytometry, the light is being utilized to identify the molecular structure, not a molecular structure, the cellular structure and volume, size, and its internal organic material, how much it is there. So that is about flow cytometry. Let's go with next application, which is light activated therapy. So when you observe this flow cytometry, light is being absorbed by these tissue molecules, whichever you are actually sending it, tissues, cells, cellular bodies, it is actually absorbing it. Then after understanding this, we can actually use it for certain therapy applications. Of course, there are particular guidelines are there, but what is light activated therapy? So light activated therapy is also called photothermal therapy. This is called photothermal therapy, which means that by shining the light onto the tissue, you are going to create some heating effects. That is called photothermal therapy. So light activated therapy is also called photothermal therapy. So this photothermal therapy uses a drug that is activated by light called photosensitizer. So it is not just merely you are using simply light is falling onto the skin and then skin will react to it. It is not like that. We are actually having some photosensitizer molecules inside which will absorb the light abruptly, and then it will actually cause heating effect. So this photosensitizing agents has been utilized for killing cancer therapy. For example, skin cancer, most of the skin cancer people are actually going with the phototherapy, where they apply certain gel or maybe they inject certain particular molecules, the photosensitizer molecules, which is very specific to the skin allergic or maybe skin cancer elements. Then these photosensitizer molecules are actually going and sitting onto the cell body. After that, you are going to irradiate your light onto that particular body. Then it photosensitizers will get heated up and then it will kill the cell. So the light can come from the laser or other sources such as LEDs also. Photothermal can be also be utilized, called PDT. So various light frequencies started from UV to IR, ultraviolet rays to regular IR rays also we can utilize. There are many applications are there. Will be utilized for exciting the cellular material for intended purposes, which can be either supportive in nature or destructive in nature. Sometimes this phototherapy is to improve the cell culture. Sometimes the phototherapy can be given to improve the cell culture, that is supportive in nature. Sometimes we wanted to destroy the cell. For example, cancerous tissues are there. We wanted to destroy the cell. So supportive or destructive purposes, one can use it. So let's look at the applications of light therapy. So what are the various lights that we are generally using for phototherapy applications? Let's take about UV, generally ultraviolet rays, which are less than 300 nanometers. So when you are using this ultraviolet rays, how many of you really know that these mosquito catchers or maybe the fly catchers, people are using the UV lamps. So what is the purpose of UV lamp? UV lamp is attracting the mosquitoes. So the small organs, whichever is a very small organ, for example mosquitoes or any other insects and all, they're very attracted to the UV. It is not like that. The biological systems are also having huge orientation to a UV absorption. So if you use this UV light in medical purposes, for example, it will improve the flow of blood and lumpies. For example, in any vein system, maybe there is a lump is happening. So if you irradiate with UV lamp, then there the lumpy can be removed and then improves the blood flow. And it has antibacterial effect. For example, any bacteria content is there, generally E. coli bacteria or so many. You see, UV treatment is using for our water filtration. where antibacterial, which means it is killing the bacteria inside the water. So 260 to 280 nanometers light is very good for bacteria killing, because bacteria absorbs highest amount of energy from 270 to 80 nanometers light. So because of that highest amount of energy absorption, bacteria gets killed out. The cellular body cannot sustain 280 to 70 nanometers of light. So antibacterial effect also, where your UV treated water, that is what we are drinking from our nearby plants and all. So now you see that production of vitamin D. So people used to ask you to stand in the sun in the morning, early morning. You just go stand in the sun. What is the purpose? You wanted to absorb the sun rays. So the sun rays, slight amount is contains having UV radiation, ultraviolet radiation is there, but exposure to the UV radiation for a long time will also having a problem. So only for a few minutes, like few hours, let's say less than 10 minutes up to 15 minutes, if you stand in front in a sun, in the early morning sun, you generally get UV light which has been irradiated on your skin, and then vitamin D can be enhanced. And next one, bone healing. So sometimes the soft bones, for example, you see the soft bones in the ear, soft bones inside your palm. So there are certain soft bones are there. These soft bones can be improved by bone healing technique. So next one is the IR, where we are going to use IR. IR is important for heating application. Generally, we have using for when you are using the infrared radiation, that actually creates a vibration in the molecules. The molecules are actually vibrating. There are certain vibrations are actually happening. So this IR heating, when you are actually giving this IR energy, it is created heating effects and relaxing muscles. So when you are actually treated with IR, your muscles get relaxed, because your muscles are actually the internal cellular organs that carbon bonds or carbon oxygen bonds are actually vibrating, and metabolism will be increased. So dilation of blood vessels also can be done using IR radiation. When you use IR radiation, you can have a dilation of blood vessels and increase blood flow and increase metabolism and soothe nerves. So there are very, very important applications are there. So what is the purpose of white light? Generally we see in our any malls and all, for relaxation of pain, for example back pain or neck pain, generally they use white light and then irradiate the white light onto your back of the neck, and then you get to see that there is a relaxation happening. And other examples, like using the blue light for improves the skin tone, and using the red light improves the dry skin, creates heat. Actually red light, when you're using more exposure for the skin, it creates heating effect, so that also helpful. So there are various types of light. It's been used for different medical purpose, medical therapy purposes. So you see that there are examples. Let's look at the photodynamic therapy. So what is it actually? It is for killing cancerous cells. How do they actually do? So the person will be injected, the person with cancerous receives a drug called photosensitizer. So he will be consuming or maybe injected a photosensitizer drug, which is under the prescription of a medical professional. After that, it will be taking 24 to 30, I mean, like one day to 3 days it will take, and all these molecules are actually going and attaching to the cells which are cancerous in nature, which is very specific to the molecule that you've been injected. So after that, cancer cells that absorb the photosensitizer are exposed to the light. So now these photosensitizers are already stick to the cancer cell. Now you are actually irradiating with the light, either a UV or IR. It is whatever is been prescribed for that particular application. We are going to irradiate it. After, what will happen when you are irradiating it? These photosensitizers are started heating up, and it is actually creating the destruction of cancer cell. The cancer cell will be destroyed, and light causes photosensitizers to make a form of oxygen that kills cancer cells. So this is one photodynamic therapy example. Hope you understand. So let's look at the benefits of photothermal therapy and drawbacks about photothermal therapy. So benefits of photothermal therapy is, photothermal therapy limits damage to healthy tissues, because the photosensitizers tend to build up in abnormal cells, and then light is focused directly on them. So this is one benefit. So you are not irradiating the entire body, you are just injecting the photosensitizers which are actually specific to the cancerous tissue, and then only you are focusing onto the cancerous tissue and killing them. So that is one good example. The benefit we have. And second one is the photo events. And what is the drawback? For a drawback of, or maybe I could say limitations of it. So photodynamic therapy can harm normal cells in the treatment area and cause side effects. So what are the side effects? If you really don't know how to use the photothermal therapy and it is overexposed conditions are also possible. So it has to be done under the supervision of a doctor. So the light used in the photodynamic therapy can't pass through more than about 1/3 of inch of a tissue. So photothermal therapy, you can't go with the deeper tissues. It will be only super tissues you can actually go with the therapy, but not deeper inside. So photothermal therapy cannot only be used to treat tumors that are just under the skin or on the lining of the internal organs or cavity. So inside organs you cannot actually do, only for the skin layer, the first layer you can actually treat it. So because the light cannot reach very far into the large tumors, and photodynamic therapy is less helpful in treating them. So these are the limitations that we have. So hope you understand the two of them, photothermal therapy and light therapy. One is the flow cytometry. That is one good example where the light is being utilized to understand the amount of size, volume, and amount of organic content inside. For example, proteins, DNAs, and all. That is one good application where light is being utilized. And second example is where you have a cancerous tissues and we wanted to kill them, where you are using the photosensitizers which will absorb the light and then creating the heating effects and killing the cancerous cells. And sometimes we are using for helping the blood flow to be improved, bone marrow, soft bones to be improved, nerves to be stimulated. So these are all other things that we are actually utilizing it. Hope you understand the class. Is it clear? So is it clear? Students, are you? I'm done. Alvin? Alvin sir. Thank you. Thank you very much. Thank you very much. So should I leave now? Thank you. Thank you. Thank you.